The project FreezingBioprotector, financed by the European Union Horizon 2020, aims to understand the possible role of beneficial bacteria in cold stress tolerance of plants. Climate change extreme weather events such as early spring frost have increased in frequency and intensity in the past decade. Cold stress is one of the main limiting factors affecting fruit crop production worldwide. Current methods used by farmers to mitigate cold damages include mainly physical and cultural approaches, such as overhead water irrigation, wind machines, and giant candles, which have several limitations in terms of cost, efficacy, and environmental impact. The use of natural plant beneficial bacteria could be an environmentally friendly approach against cold damage. To pursue our aim, we began a sampling campaign to collect 3 wild cold-adapted rosaceae plants from 7 different sites in Italy. To identify potential bacterial groups for cold tolerance, we characterize the bacterial community structure inside the tissues of these plants by using 16S amplicon sequencing. To recover culturable cold-adapted bacteria, we used the classic dilution plating method, and about 700 isolates were successfully obtained. Taking the results from both approaches, representative isolates belonging to different bacterial groups were chosen to identify potential isolates for enhancing cold stress tolerance in plants. For this purpose, we developed a high-throughput screening method with in vitro strawberry seedlings. To determine the protective abilities of such isolates, we used electrolyte leakage, which measures the ions released from the plant cells due to the damages to the cell membrane. A major result include the identification of 15 isolates with promising protection level against cold stress. These isolates are currently being tested for their ability to improve freezing tolerance of micropropagated and potted apple plants. The project has scientific impacts on the deep characterization of bacterial contribution to the cold tolerance of crops, and a positive impact on society by increasing the quality and quantity of apple crops by improving their cold tolerance. The project has also impacts on the economy by contributing to the development of new bacterial-based products for the cold protection of apple crops.